Hello, welcome to my channel Correct Biology. My name is Dr. Vikash Bhardwaj, and in this video I am going to teach you the difference between prokaryotic and eukaryotic cell. So here within this video you are going to learn the difference between prokaryotes and eukaryotes. Now try to see here pro. Here pro means primitive. Here U means true. And let's see this term karyotic. Here it is going to means nucleus. Now here it means nucleus. Now in the case of prokaryotic cell, remember what you are going to see: primitive type nucleus. You are going to see primitive type nucleus. Here if you see in the case of bacteria, what you are going to see, DNA, it is present within a region, and this DNA, it is not being surrounded by membrane. So here it is present within a region not surrounded by any membrane. So here you are going to see primitive type nucleus. Remember, in case of prokaryotic cell, in actually the nucleus is absent. Here this region, it is known as nucleoid. This is known as nucleoid. Keep in mind, in the case of prokaryotic cell, nucleus is absent. Now here if you see in the case of eukaryotic cell, what you are going to see, you are going to see a membrane bound nucleus. True nucleus is present within the prokaryotic cell. So here if you see within the eukaryotic cell, what you are going to see, you are going to see true nucleus. So here around the nucleus you are going to have nuclear membrane, or you can say nuclear envelope. Okay, so this is the difference, one major difference. In case of prokaryotic cell, nucleus is absent. Here what you are going to see, nucleoid. In case of eukaryotic cell, what you are going to see, membrane bound true nucleus. Now here I just want to tell you the examples of prokaryotic cell. Example of prokaryotes, what are? Bacteria and archaea. Okay, so here keep in mind the bacteria and archaea, they are the example of prokaryotic cell. Same way if you see the examples of eukaryotes, it includes here the fungi. Okay, then you are going to have plants, animals. Okay, these are examples of eukaryotes. Here you are also going to see protozoa. Okay, these are also the examples of eukaryotes. Now try to see here bacteria, archaea. Okay, these are unicellular organisms. Okay, and here if you see fungi, plants, animals, protozoa. In case of fungi, if you see yeast is unicellular. Okay, so protozoa, they are unicellular. So here you can see both unicellular as well as multicellular organisms. So keep in mind, in case of prokaryotes, what you are going to see, unicellular organisms, but in case of eukaryotes, you are going to see both unicellular as well as multicellular organisms. Now another important difference, keep in mind, in case of prokaryotic cell, okay, you are not going to see any compartmentalization. In the case of prokaryotic cell, you are not going to see any compartmentalization, while in the case of eukaryotic cell, what you are going to see? Compartmentalization. What it means? Try to see here. If you look into the size of the prokaryotic cell, generally these are small. Let's suppose this is one bacteria. Okay, here if you see the size, it is 1 to 2 micrometer. But if you see the size of eukaryotic cell, okay, here I have drawn this, you can say the animal cell. Generally the size is 10 to 100 micrometer. Comparatively, if you see the eukaryotic cell, it is going to have large volume, large space. So here what you are going to see, compartmentalization, means within the eukaryotic cell you are going to have other membrane bound organelles. Like here I have mentioned, here you are going to see the presence of nucleus. Here you are going to see around the nucleus endoplasmic reticulum. Then you are going to have Golgi bodies. Then here you are going to have presence of mitochondria. Okay, and if it is a plant cell, here you can say there is also presence of chloroplast. Now try to see here, in case of prokaryotic cell, what you are going to see? Compartmentalization. Sorry, no compartmentalization. While in the case of eukaryotic cell, you are going to see compartmentalization. Here the organelles, they are present. So in case of eukaryotic cell, organelles, they are present, while in the case of prokaryotic cell, organelles, they are absent. Now here I also want to tell you, if you see in the case of prokaryotic cell, in the case of prokaryotic cell, what you are going to see? The ribosomes. Okay, ribosomes, they are of 70S type. So here S stand for Svedberg unit. Okay, so when you are going to centrifuge the ribosomes of prokaryotic cell, generally they are going to sediment at 70S. And remember, the ribosomes, they are going to have two subunits. One is large, another is small. So here in case of prokaryotic cell, the ribosomes, they are of 70S type, and here it is going to have two subunits. One is large subunit. Here the large subunit is 50S type, and the small subunit is 30S type. And if you see the case of eukaryotic cell, what you are going to see here, in the case of eukaryotes, the ribosomes, they are of 80S type. They are of 80S type. Here also you are going to have one large subunit, another small subunit, and here if you see, the large subunit is of 60S type and small subunit is of 40S type. Remember, this is Svedberg unit of centrifugation. Now here another important difference I just want to tell you, in case of prokaryotic cell, generally what you are going to see, the cell, they are going to divide by the process of binary fission. Okay, one cell will simply divide into two by the process of binary fission. Yes, we will discuss in detail the steps of binary fission later. But here if you see, in the case of eukaryotic cell, okay, you are going to have generally, okay, the advanced mode of cell division process, that is known as mitosis. Okay, so this is also one major difference. Now another important difference, what you are going to see, in the case of prokaryotic cell, okay, what we are going to see, please correct it. Okay, in the case of prokaryotic cell, what you are going to see, that this is the cell, okay, and here the genetic material, it is present necked, okay, it is present necked, and it is in direct contact with the cytoplasm. So here this is area is cytoplasm, and here you are going to have the DNA which is present within the nucleoid region. Now try to see here, at the site where there is DNA is present, the process of transcription is going to occur. What is transcription? Transcription is synthesis of RNA. From DNA, what is going to happen? RNA will be synthesized. So let's suppose this is the RNA being synthesized. Now this RNA, okay, in case of prokaryotes, it will be captured by the protein synthesis machinery. Okay, so in case of prokaryotes, what you are going to see, the process of transcription as well as process of translation. So now here this RNA, let's suppose this is messenger RNA, it will be used as a template by the ribosomes, okay, and let's suppose it is going to synthesize some protein, okay, it is going to code for some protein. So here this process is known as a translation. So here you can see transcription and translation, okay, they are coupled process, they are coupled process, okay. But here I just want to tell you, in the case of eukaryotic cell, in the case of eukaryotic cell, what you are going to see, that this is the cell, and here you are going to have the nucleus. Within the nucleus you are going to have the DNA, okay. So here transcription, okay, the transcription process, it is going to occur within the nucleus, while the process of protein synthesis, translation, it is going to occur within the cytoplasm. So here what is going to happen, RNA will be synthesized, then later it will be transported, and here you see the process of protein synthesis will be carried out, okay. So here you can see transcription and translation, they are going to occur in different compartment. So here in case of eukaryotic cell, remember transcription and translation, they are not coupled, okay. So there are more difference, but in this video I have told you the major difference between prokaryotic and eukaryotic cell. Please do like, share the video, and subscribe to my channel Correct Biology. Thank you. Have a nice time.